Golden Jubilee lecture series. Today we have a very eminent.
Sir, you have muted, sir. Mondal, sir.
No, he has disconnected. He will come back.
Unmute. Unmute. Dr. Mondal, unmute. Dr. Mondal, unmute.
Oh, yeah,
He's not there.
Acha, disconnect.
Yeah, he has disconnected.
Sorry, I got disconnected due to some other issues. So it's a great pleasure for us that we had with us Professor Wayne Michael Jurick too, and he's an eminent research, scientist in the field of Postharvest plant pathology. And it's a great pleasure, Professor Wayne, that you have kindly consented to us and to have such kind of, you can say, lectures series. What is the background? You know, I think the IPS is celebrating its Golden Jubilee celebrations in this year, and that's why we proposed a series of lecture series. And I think we really honored to have your lecture in this series, and we are really thankful for your kind consent. And I think all of us will be benefited and enriched with your rich insight into this post-harvest plant pathology in particular. So without any delay, I once again welcome to all our President, Pratibha Madam, our Secretary, Dr. Ravin Gogoi, and also eminent Madam Sangeeta, Prashant Jambulkar, Sridhar sir, Dilip Lakshman sir, and Pramod, and all the others. So that we are really great that it is a great evening from India and good morning from their side. So without any delay, I just welcome you, Professor Wayne, to start your lecture. Professor Wayne, sir.
Thank you, Mondal, sir. Now I request Dr. Prashant Jambulkar, sir, to introduce the speaker to us.
Okay.
Thank you.
Thank you, Dr. Sangeeta. Good morning. First, I have told you that it's for you good morning from our place. Dr. Wayne Michael Jurick is most welcome to you, sir, on this Platinum Jubilee lecture series of Indian Phytopathological Society, which is based in New Delhi, India. And we are delighted to have you here today for this lecture. Dr. Wayne is a lead scientist and research plant pathologist at USDA ARS Food Quality Laboratory, Beltsville Agricultural Research Center, Maryland, USA. He pursued his BS, MS, and PhD in plant pathology from University of Florida. He joined USDA ARS Beltsville, Maryland as a postdoctoral research associate in 2008, and later he became research plant pathologist and rose to a position of lead scientist and research plant pathologist in Food Quality Laboratory. He is a reviewer and editor of very prestigious journals like Phytopathology, Phytofrontiers, and Frontiers in Fungal Biology. He is an outstanding researcher. His research articles are among the top 5 and viewed mycology articles in PRJ published, which is published in 2019, and it has as many as 2029 views. Another distinguished achievement in his research was highlighted in President's budget request to the Congress for the financial year 2020. That's a great achievement. Congratulations to you, sir. He is a prolific writer and he has published many excellent research papers which has been highly reputed, published in highly reputed journals in the field of plant pathology. With this illustrious academic as well as scientific background to you, Dr. Wayne, we are very much excited to listen to you. Platform is yours. On behalf of IPS, I request you to deliver your lectures. Please.
Well, thank you very much. Can everybody hear me okay?
Yeah.
All right, very good. I'm going to share my screen here.
Yeah, please.
And get going. All right, let's see.
And I must tell you that there are many participants who are joining to you by YouTube link also.
All right. So far, so good. Everybody see the screen?
Yeah, it's visible, sir.
Yes.
Yes, it's okay.
Excellent. All right. I really appreciate the introduction, the kind introduction, and the opportunity to speak today. I'm excited about this opportunity. Again, my name is Wayne Jurick. I'm the lead scientist and research plant pathologist in the Food Quality Laboratory in Beltsville, Maryland, at the Beltsville Agricultural Research Center. And the title of my talk today is entitled Tools of the Trade. Blistering 1 modulates multiple aspects of Penicillium expansum virulence during post-harvest decay. So I'd like to start my talks with a brief outline to give you some idea of how the talk is organized and to let you know what's coming up. So first I'll introduce you to blue mold, the disease, and also the pathogen that causes blue mold of pome fruit. So a little bit about the biology of this thing. Second, we'll talk about some Tdna mutagenesis efforts to uncover some virulence loci that are involved in apple decay and the fungus. Then we'll get into more of the meat of the project, which entitles the Blistering 1 multiomics, multiomics is just the integration of multiple omics technologies into one study. Okay, and then finally, I'll end with a summary and some conclusions. And my hope for you today is that you guys will learn some new information about post-harvest, especially apple, and also give you some more insights mechanistically into what's going on with the fungus and how this thing utilizes very intricate, specialized mechanisms to cause decay and produce toxins, which is the crux of the story today. And as we go along, if you have questions, go ahead and put them in the chat, or I guess we'll field them at the end, I believe. So anyway, here we go. So to get started, post-harvest decay, as it's defined, happens after the fruit are harvested. So apples generally are stored in cold storage for multiple months on a time, sometimes up to a year in specialized storage. And you get blue mold. It's caused primarily by Penicillium expansum. Now, there are multiple species that cause blue mold, but the most predominant and the most globally widespread is expansum. It's very virulent. And as I said, blue mold occurs on pome fruit. Penicillium infects both apple, pear, quince, also medlar, if you're familiar with that, and after they are harvested, when the fruit are in storage. Now, in the US, I'm speaking primarily, losses in storage range from 1 to 5%, which is pretty low for fungicide-treated fruit. However, those losses are significant because there are significant economic costs associated with repacking, and also they risk rejection from particular producers for that level of decay. So the tolerances are quite tight here for post-harvest losses. And this is under the best circumstances. In places where they're not using fungicides or the storage facilities aren't as sophisticated, you can encounter 30, upwards of 40, 50% loss in storage, depending on the variety and the time. And this blue mold and penicillium is a globally distributed pathogen with high economic and human health impact. And I'll get to that in a minute. Why is it of human health concern? But so it not only reduces fresh fruit for consumption, but it reduces their quality and costs the growers money and the packers money when the shipments get rejected. So I want to show you what the disease looks like in situ, if you will. Here's a picture of an apple. This is at a commercial storage facility in Pennsylvania, just about an hour and a half north of where I'm located. You can see the apple here in the middle is heavily colonized with penicillium. This is a typical blue mold, pretty aggressive infection. You can see the bluish green colored spores that develop on the outside of advanced decay lesion here. And this gets really mushy and musty smelling. All right. So this is in amidst all these other healthy fruit here. You have this one apple that I placed nicely right in the middle here. Now, cheating a little bit, here are inoculated, artificially inoculated European pears that are, I believe, are Bartlett's. If not, they could be Anjou. I don't remember. They look more like Bartlett. And you get the same characteristic, very distinct circular lesion here. You just start to see some fungal growth and conidiation occurring at the center of the lesion. And this thing will continually expand outward until the fruit is completely engulfed. And the same story occurs with Asian pear. I don't have any good pictures of quince or medlar with decay, but Asian pear is the same story. You'll see, this is manually inoculated with conidial suspension in the center. And this is about 4 or 5 days after inoculation. You get this nice, very perfectly formed circle. And the decay is easily separable from the host tissue. And is very characteristic, easy to identify in storage. Okay, so this is what the disease looks like, and it's easy to spot and it's fun to work with. So if you take a closer look, if you have a scanning electron micrograph, which we have at Beltsville, you can basically take a nice cryo-EM photograph of the conidiophore and the conidia of this fungus. And you see in the background here, the mycelium. This is a really nice picture illustrating the world conidiophore, all right, or the witch's broom, if you will. It's referred to as that. And then you have these conidia that are produced terminally in chains at the top of each conidiophore, and these just go on up. And they're really nice. What happens is they break off, and then they are involved in the disease cycle. If you take a closer look, this is a really high magnification. I believe this is 25,000x. You can see the spores here that are produced in chains, almost like a balloon that you've taken and twisted and knotted together here. I mean, you can see the connections between the spores. So these are really interesting close-up shots of a typical Penicillium expansum. Even other penicilliums, you'll see this world conidiophore and these terminal conidia, which are the primary source of inoculum for disease in the storage facilities. Okay, so a little bit about infection biology. This fungus is aggressive. However, it's not capable of direct infection. So it requires a wound for ingress. And that happens a lot during the packing, storage, handling. You get bruises, you get stem punctures. You get a lot of different issues that go on that cause places for the fungus to gain ingress, okay? It can also go through natural openings like stem ends and stem pole areas. The fungus during the decay process modulates the host pH by organic acid secretion. So it lowers pH of the host even more. And these organic acids are typically all the dicarboxylic acids that you find from TCA cycle metabolism, primarily oxalate, but there's other ones. And then there's a concomitant secretion of these carbohydrate active enzymes. And one of the most famous one of that class would be the polylactorases, which are secreted and involved in tissue softening, actually tissue degradation, but there's others. There's proteases, there's cellulases, there's pectin methylesterases. So you get the secretion of these cell wall degrading enzymes. And then you have on top of this a non-host specific toxin called patulin. And this is where the human health aspect comes in as well as the virulence. So patulin is secreted. It's a toxic lactone compound as indicated here by this ball and stick structure. Chemically speaking, it has two electrophilic centers, which form adducts with cysteine and thiol groups. So in human cells, it conjugates with glutathione and drops the glutathione pools. So those are no longer able to help quench any of the oxidative stress for the cell. This thing is also, at this carbonyl function, is also prone to hydrolysis reactions, both acid and base catalyzed. And those reactions open this ring up, okay? And then that goes into the degradation pathways. You got to open the ring up. And why I talk about that as well is so this thing's not only a virulence factor for the fungus, but it's toxic to humans. And there's regulations on this toxin for the FDA in the United States and the European Union. And the other bad part about this molecule is that it's very chemically stable. It's very heat stable, survives pasteurization, and it's not completely eliminated by fermentation. So those that are using apples, cider apples, low quality apples for making alcoholic cider, you're not going to completely eliminate patulin, and it's a potential carcinogen. It's hepatotoxic. So that's an additional problem for having this fungus around, causing problems. So what I want to introduce you to here is a figure that I took from Li et al. 2019 from Environmental Microbiology, and this is from Shiping Tian's group in China. She's got a really nice group, and she went in and knocked out every single gene in the patulin biosynthetic cluster. And I don't want to overwhelm you with this, but what I like about this diagram is it shows you a couple of things. First of all, we start with acetate here, and this is a polyketide product, and it goes through 11 enzymatic steps, and there's various enzymes involved here as you move throughout the process. But if you notice, there's a transporter here at the edge of the cell, and this double bonded alcohol up here called ascladiol is pumped out. All right, and that's not toxic. They've done studies with this. That intermediate is not toxic. It's pumped out. And what you'll find from my data today, from the proteomic data, this enzyme that converts ascladiol to patulin is secreted. So we have evidence that this enzyme is moved outside the cell. So now you have the enzyme and the intermediate, and guess what? They work together, and now they form patulin, which is toxic. So this is important from a way that this fungus, when it produces this, is able to escape its own poison. So it converts this poison outside the cell, which is very important. Okay, so that I want you to remember, if anything, this secretion of this intermediate and this enzyme here. Okay, going the wrong way here. So how do you control this thing? So what do producers do? What are the options for control? Well, you can use post-harvest fungicides. There's about 3 or 4 that are labeled, but there's problems with resistance, and there are resistant isolates out there for almost all but one of the chemistries available that are labeled in the United States for blue mold for pomes. You can do some sanitation, like cleaning your orchards and sanitizing your bins and cleaning the air in your packing house, but those things are not routinely implemented due to increased costs and time. Well, what about one of the other legs of classical management in plant pathology, using resistant cultivars? Well, there aren't any. Okay, so there's no host sources of resistance in commercial apple cultivars to blue mold, yet any other rots that are causing problems in storage. So this is the rationale for why I work on Penicillium and other rots. We need to develop new tools and tactics and management strategies to control blue mold because of its economic importance and because of its potential for human impact. So with that, I'd like to say we took this Tdna mutagenesis approach. So the goal here is to use a genetics approach to create Penicillium expansum random Tdna mutants. So you incubate your fungus with agrobacterium carrying a hygromycin resistant cassette, and the agrobacterium inserts randomly its Tdna into the fungal genome, and you select these mutants on hygromycin amended media. And what we did is we screened this collection, if you will, of Penicillium for defects in apple. So we were looking for virulence phenotypes and their impact on fungal growth in vitro. And then what we did once we have those mutants is we tried to uncover their Tdna integration events, the Tdna copy number, and the impacts of the integration on gene expression. This is a pretty routine way of generating random mutants that you can now go in and use and explore looking for virulence phenotypes. So this is, I got something in the chat here. Let's see. Okay, never mind. All right. Everything good so far? Everybody hear everything? Everything clear? Slides working good?
That's good. It's well. It's going well.
It's good, interesting.
Okay, good. Okay, so an overall scheme of what we did, we generated almost, well, 456 Penicillium expansum transformants carrying the Tdna resistance marker. And we screened every one of those in Apple. And we looked for significant reduction in virulence, significant meaning 20% or more reduction in virulence. So the lesion had to be 20% or more. We ended up with 43 contenders, and we screened them again in Apple. And we also looked at their mycelial growth. So what we did is we verified our phenotype in Apple, and we did not want mutants that grew poorly in the plate or had a severe growth penalty. We wanted to try to uncouple the growth impact on the fungal growth in culture from the decay phenotype. And we were able to do that to a point, and we limited it down even further to 14. Then what you do is this very interesting technique heavy process called tail PCR to try to figure out where your Tdna inserted. So you can use some primers that flank your insert, random hexamers, and you can use some internal primers, and you can try to amplify each section of the flanks. And you get down, we got down to 6 strains. So you have to start with a lot of mutants. You got to start with a lot of transformants to get even just a small number. And with these 6, we check the copy number and their gene identity. Yeah, come on now. Now my computer's frozen. Okay, so what we have here is the identity of the 6 mutants that we generated, and then where the based on southern hybridization, I can tell you using the hygromycin cassette as a probe, we had a single insertional event in each one of these mutants. So that's the first thing. So we use Southern blot to screen these things, single integration, so that's good. And then with the tail PCR, we were able to tell what part of the gene we inserted in. We know that Agrobacterium prefers AT-rich regions, so you're going to get integrations that are predominate in the 5 or the 3' untranslated region of the genes, i.e. the regulatory portions. And that's what we see here for the most part, except for this one, this T625 mutant, which is special. That guy had a direct insertion into the coding region, and I'll talk about that more. So we wanted to gain some insight into what these genes do, and unfortunately, a lot of these are hypothetical. So hypothetical protein 3, 2, 1, no putative domains detected on some of these. This one particular mutant was a glucosidum N, a NAG transferase involved in decorating proteins. Has a protein domain here, GPI domain. This hypothetical has a glyoxylase and an ATP synthase. We have a putative heat shock protein with a DNA J domain and a hypothetical protein. So what I want to call your attention to here is that 588 and 711 integrated independently into the same locus. So they're essentially the same mutant, okay? And the integrations, as I said, occurred in the 5' and 3' UTR. And based on all this, I was looking and I was trying to think, okay, now which one of these am I going to use for further characterization? And the fact that we have an insertion in the coding region, and we have a very interesting gene here with a heat shock protein DNA J domain. That was the mutant that I selected for further genetic, biological, and proteomic analysis. Okay, so what does this thing look like in apple? Well, the apple inoculated on the left is inoculated with the T625 mutant. The apple inoculated on the right is the wild type parent, and you can see there's quite a difference here. Still pathogenic, but it's much reduced. And the mean lesion diameter here, if you measure this, is much smaller. Here's the wild type, R19 wild type. Here's the mutant, and they're statistically significant. They're specifically 6.6 millimeters smaller than wild type. So not a very distinct phenotype in apple, but it's significant. And by the way, this work that I'm presenting today is all published in Molecular and Cellular Proteomics in 2020. And if anybody's interested in the reprint, I can send it to you, because with that journal, we had to, I don't believe that journal will, I think you have to pay to get the reprint. So I can send you the reprint if you're interested. All right, so we have a mutant with a good virulence phenotype in apple. One of the first things we did was check conidial germination rate, because we thought maybe the conidia germinates slower. That's not the case. I'm not showing you that data, but there's no difference between the mutant and the wild type. They both germinate at the same rate and the same level. If you look at the mutant in culture, remember I said that you want to uncouple fungal growth in the plate from as much as you can from the virulence phenotype. The mutant is on the left, and the wild type is on the right. And what you'll see is data here on the right with this bar graph. R19 is a wild type, ends up going about 6 millimeters a day, whereas our hygromycin resistant mutant here's on hygromycin, not on hygromycin, doesn't matter. Grows a little slower, significantly different here. It's reduced by about 1.4 millimeters per day. So there is a little bit of a growth penalty. And you can see that here in these colonies. You can see the size. But morphologically, it's very similar, all right? Not much phenotype in culture. If you look at the actual locus that the Tdna jumped into, there's a line diagram here. We have a single locus called, we're naming it blistering 1. This is the Tdna here, Tdna with encoding the hygromycin. And it jumped into the coding region about 137 base pairs downstream from the putative start site. The gene is small, has one domain there, the DNAJ domain, which if anybody knows what DNAJ domains do, they're involved in protein-protein interactions, specifically with other proteins that help fold, okay? There's no introns in this thing. It's an intronless gene. And we have a single site integration confirmed not only by PCR, but Southern lacks, I just said all this, lacks introns. If you look at the expression of blistering 1 locus in vitro, so on plates, you just take a mycelial scrape and you look, the gene is expressed in the wild type pretty highly here, but in the mutants, the expression is undetectable, which is good. And then if you look in apple, yeah, it's expressed in apple and wild type as well, less than in culture, but you also can't pick it up in apple either. So we have an essential null mutant here, okay? Even though we still have the intact copy of the gene, this thing is not being expressed or is not detectable in its expression, which we liked. If you take a closer look using cryoscanning electron microscopy in the mutant, what you'll find is the typical world conidiaphores again, with the conidia being born in chains. But something very odd here is the appearance of these blisters and bumps all over the main hyphae, okay? And what that, we don't know what that material is. We hypothesize it could be proteins that are missecreted. We don't know, but if you compare that to wild type's completely smooth. Okay, so we've named this mutant based on this phenotype, this electron microscopy phenotype, blistering 1, because it looks like sort of a hot dog. If you cook it too long, it gets all this blisters on top. So we named it mutant after that. All right, we got a real good imagination here. All right, so if you take a closer look, Joe Mowry did some really nice transmission electron microscopy here. And this phenotype is not just detectable from the outside. There's some internal issues going on here. So let's take this diagram or this electron microscopy photo piece by piece. The top half here, all 4 figures in the top are all wild type. So if you start with A, You have a nice round cross section of a hyphae, and you have a nice thin cell wall, and you have a really compact structure. Longitudinal sections, everything looks normal. You see the cell wall here, and then the tip, the growing tip, you have the accumulation of the vesicles at the growing part of the fungus, which is what you'd expect. In stark contrast, your blistering 1 mutant, look at how lumpy and bumpy this thing is on the outside, and you can see how thick this cell wall is. Joe actually measured that, and the cell wall for R19 is about 116 nanometers. And the one for T625, which is your blistering 1 mutant, that's almost double. All right. So you got a thick cell wall, it's bumpy, and you have these big pockets, or air in botany, they'll call them air lacuna, some kind of huge air spaces in here that are apparent in the longitudinal sections. Here's a close-up of the cell wall, and you can see here to here, the difference from here to here, much thicker. And there's some dysregulation with the vesicles here. There's still a tip-high accumulation, but there's more spread back here subapically. So there's some very interesting phenotype at the microscopy level with this mutant. But we wanted to go beyond just looking at the fungus with the microscope, and we wanted to use a comparative proteomic approach to try to understand mechanistically what's going on with the virulence penalty, and why is this thing not able to cause decay like wild type. So we used fungal cultures. We grew them up, wild type and mutant, in a defined medium containing apple tissue as the carbon source. And we basically sought out to identify and quantify secreted proteins in the apple medium. So basically to characterize the secretome, or all the proteins that are secreted by the fungus. And then likewise, elucidate global changes in the proteins from the fungal mycelium. So looking at what I call the mycelium, all the proteins in the fungal mycelium that are going on, and compare the wild type with the blistering 1 mutant. And the way we chose to approach that was with this TMT proteomics platform. So as I said, grow the fungus up in apple liquid shake culture for 3 days, harvest the mycelium and the broth, flash freeze them and store them at -80, and then purify the proteins and digest with trypsin. At that point, you label the peptides with tandem mass tags and run on the Lumos mass spectrometer. And the Lumos, so here this is depicted pictorially, and Brett Cooper and Wes Garrett did this work with the tandem mass tags, MultiProteomics. And what you have is this, your labeled peptide fragments here, and your MS1 scan identifies your precursor ion. Your MS2 scan is where your peptide sequencing comes from. And then your quantitative aspect here comes in from your MS3 scan, where your reporter ion is actually quantified. So you can do quantitative and qualitative proteomics here. So it's very informative, very sensitive technique. So if you look at the broth from the liquid cultures and wild type versus mutant, you're going to find some pretty interesting things here. So what we did is, I'm not showing you everything because the list, it's a huge list, but these are like the top 10. These are proteins that are absent in the mutant, but present in the wild type, or reduced more than 1 log 2-fold compared to wild type. So these are what I call class ditchers. All right. These are your kids that don't go to school. They should be there, but they're not. Okay. So I call them class ditchers. So if you see these classes, so what are these enzymes, or what are these proteins? All right. Well, they're a whole bunch of cell wall degrading enzymes: xylosidase, glucosyl hydrolase, alpha amylase. Look at this. Remember PAT-E? PAT-E is secreted. That's one of the patulin biosynthetic genes. There's PAT-B, PAT-O, pectin methylesterase. Then there's a whole host of these other oxidases, polygalactoranases. There's even a sugar transporter. these are all significantly downregulated, all right? So there's some interesting things going on here in the mutant, all right? Also, we looked at proteins that are absent in the wild type secreted fraction, but found in the mutant or reduced more than 1 log 2-fold compared to the mutant, all right? Now, these are people that I compare these proteins to people that show up to parties that you don't want them to be there. I call them the party crashers, all right? So what we have here, so what are these? What are the ones that are absent? These are chitinases, glucanases, more cell wall degrading enzymes. There's some rodlet peptides here, hydrophobins, cholinesterases, so lots of enzymes. And what this really shows you here is that there's global misregulation of the secreted proteins in this mutant, okay? So this thing can't secrete the proteins properly. It's messed up, all right? And so we wanted to test that. So we led with this hypothesis that, well, maybe the fungus, it's not secreting the proper enzymes, so it can't macerate as well. So then it can't cause the decay like the wild type. So we take apple tissue and we take spent broth from each the mutant and the wild type, and you incubate apple cores at a set weight in this spent broth, and then you measure weight after a certain amount of time. And what you'll find here is that the wild type broth, this was a plug, literally a plug of tissue, and it was very amorphous. It had lost a lot of size and shape. Well, look at the mutant. All right, it's a little bit different, but it's not much. Not much change has occurred. And then the negative control, which is buffer only, is completely unchanged. And quantitatively, if you measure mass change, you'd expect your wild type would have the most change, which it does here, whereas the mutant doesn't have as much. It's all, very much, it's less, and the negative control, almost none. So this is mocking exactly what happens in the fruit. So we had a little bioassay that we could basically prove our proteomic data, that the spent broth macerates apple tissue better than the mutant broth, all right? So that's pretty cool. We validated our proteomic findings. Taking it a step further, knowing what's involved in virulence, right? So we looked at patulin. We used HPLC, look at patulin concentration in the wild type again, and the mutant. Look at the difference. Almost undetectable, not quite, but almost undetectable in the mutant, which was pretty interesting. So the patulin levels are greatly reduced. And what else do we know? This fungus is supposed to be able to acidify its environment. So it's supposed to dump acid. Well, R19, the culture pH is around 5.2 here after X number of days. However, the mutant, I believe this is 3 days, the mutant allows the pH to creep up. So not only do you not secrete the proper enzymes, then you don't put them in the right environment to function, okay? So these are all making sense why our mutant is impaired in its ability to cause optimal levels of virulence here. And further exploring this explanation, so why is patulin down? Why can't we detect it? So we have the power of looking at every single gene or protein, if you will, in the patulin biosynthetic cluster. Remember, I showed you that diagram with them all located as enzymes around. Well, here you have them annotated from A to O. You have their localization. Some are cell membrane bound, some are ER, some are secreted, and you have their function. They're all different enzymes, transporters. All right. The bottom line here is the reason why we don't find patulin is 10 of the 15 patulin proteins are down in the mutant. I mean, look at this. Down negative 2.4, down negative 2.8. And in some of them we couldn't find, only 2. But more importantly, look at Pat E, significantly down in the mutant, targeted extracellular. All right, that converts that last step. So this explains or helps explain why our patulin is nearly undetectable in our mutant. These things aren't completely shut off, but they're pretty well inhibited as far as being produced at the enzyme level. All right, so before I go any further, so we know right now our secretome is altered in the mutant. So what we looked at, as I talked about earlier, is we wanted to look at the mycelium, or all the proteins in the mycelium in the mutant versus wild type that could be altered. And our original hypothesis was that, well, maybe there's not something that's so complicated. The simplest model would suggest that these proteins are just backing up in the mutant. They're just not getting out. And what we did is we took a global look, first of all, at our proteins that are up in the mutant. All right. So what you'll see here on the left diagram is a pie chart and the predicted cellular location of each one of these, of all the proteins we detected. And what you'll see, there's global shifts in the mutant. What's up in the mutant is on the left, and what's down in the mutant is on the right. And what I want to call your attention to here is three areas: the endoplasmic reticulum, all right, the nucleus, which is here, and then the extracellular fraction, which is down here. All right. So there are major changes in the ER, the nucleus, and the extracellular. A lot of these other categories are different, but they're moderately altered. So what you can do with your proteomics data is you can load all that into the KEGG mapping system, which basically puts them into different biochemical categories and assigns function, and you can tell what's upregulated, what's downregulated at the protein level. So that's what we did here with the mycelial protein. So we switched to the mycelium now. We're no longer in the secreted fraction. This is a very complicated diagram, and it tends to overwhelm, but I'll attempt to simplify this, all right? This is just a KEGG map, and where you see color here, yellow is down, okay, and blue means up, all right? And this is the diagram of a yeast cell from the KEGG map for protein secretion. And what I was expecting is to see, based on my very simplistic model, is that, oh, all the proteins are just getting trapped in the cell, and therefore you would all see them all yellow, all down. But that's not what you see. You see some are yellow, some are blue, and basically the short story here is that the fungus is trying to adapt and trying to make up for this fact that it can't secrete properly. All right. So in the interest of time, I'm not showing you all the KEGG maps, but we KEGG map the secretion pathway. We KEGG mapped endocytosis, in other words, protein export and import. We looked at nuclear proteins. This same pattern exists for all these other pathways. So what I want to just make sure I drill home here is that there's a global misregulation of protein secretion mechanisms here that are involved or responsible for the misregulation of the proteins being secreted into the medium. This is all due to one gene, and I don't remember if I said it, but blistering 1 is a single copy gene. All right, there's only one copy of this thing in the genome. So how am I doing on time? All right, I'm doing good. Summary of what I have so far would be that blistering 1 is a single copy regulator of fungal virulence and Penicillium expansum. I think that's pretty obvious based on what I showed you today. The mutant exhibited misregulated carbohydrate active enzymes secretion, reduced patulin, and could not effectively alter its ambient pH and culture. The patulin gene cluster is disabled in the mutant, which gives us the clue, or gives us the result that this is the nearly undetectable patulin in the broth. All right, it can't make it properly. So in a more global sense, we see that there's changes in protein secretion, nuclear and endocytosis related proteins that are altered in the mutant versus wild type. And these machineries for protein trafficking are changed in the mutant. Okay, so there's a lot of proteins, and I didn't go into all of them, but the sec proteins and things involved, the vesicle trafficking, those are altered. And so our hypothesis now at this point, after all this time, is that blistering 1 forms complexes with fungal protein sorting machineries to ensure that these proteins that are destined for secretion are folded, decorated, and moved out of the cell properly. That's the hypothesis, all right? So what is the next step? All right, so we blew through this study. We worked on this. We published it. We were moving down the line, and our next step here was to generate multiple blistering 1 deletion mutants and assess their phenotype in vitro and their impact on apple fruit virulence. So the main weakness of my study here is that I have 1 mutant. All right, I have 1 mutant that I characterized all this on. So we wanted to take a reverse genetics approach now that we know the locus and go in and delete the entire locus from the genome and see what the phenotype was. All right, see if we can mimic that apple fruit virulence phenotype. And then we wanted to complement the deletion mutant with a GFP tag blistering 1 locus, determine where blistering 1's hanging out in the cell. Where's it located? What's it doing? And then once you have a GFP tagged in your protein, then you can use antibodies to that GFP to do pull downs and interaction studies to understand what this thing is doing and test your hypothesis. If blistering 1 is interacting, what is it interacting with, and is it interacting with other proteins? So we continued to move and hammer on this blistering 1 story, and we did work with Nancy Keller in Wisconsin, who is Aspergillus penicillium expert and really has this transformation system up and running, very highly efficient here. So we did they did the transformation and basically knocked out blistering 1. So you're looking for a loss of this band here. And you're looking, this is the positive control to make sure that your template's good. There's 14 different transformants here, 13 of which are all deletions, which are great. Very high efficiency. The southern blot here, we picked a wild type, and we picked the first 5 transformants. You expect 2 fragments. You use an enzyme that cuts in the middle of your marker. You have a probe that spans the marker, and you get the expected pattern in T1, 3, 4, and 5. T2 must have had another integration someplace. You got an ectopic. You must giving you an extra set of bands, so we stayed away from this mutant. So now we've generated 4 independent knockouts, clean deletion knockouts of blistering 1. And again, with the southern blot, you'd expect a wild type 1 band. With a knockout, you'd get 2. So that's what you want. So first thing I did when I got my hands on all these knockouts was I took my original wild type, R19, I took blistering 1, and then I took the parent and the control strain for all my blistering 1 deletions. So wild type, blistering 1, wild type, control strain, and then these are all independent knockouts. And I looked at conidial germination under the microscope. And as you can see, much to my happiness here, is that you have virtually no difference in conidial germination rates, which is the exact same thing we saw with the Tdna mutant. That was good news. However, the only difference between the 15-1 deletion strains and my Tdna strain here was that early on in the germination process, the mutant has a very distinct phenotype in its elongation of the germ tube. So it starts to come club shaped a little bit, and then it straightens out and catches up. So there's something going on there that's very distinct from the Tdna mutants. Okay, but otherwise these things germinate at the same rate. If you put them in apple, all right. So since all my mutants were relatively similar, I picked T1. I used that and I inoculated what is this? Honeycrisp, everybody's favorite apple, Golden Delicious. Gala and Golden Delicious. And you can see here, again, the subtle defect in lesion size, where the mutant is less than wild type. It is subtle, but it is statistically significant. And so we've replicated, or we've shown that our mutant is, our deletion mutant has a very similar, almost identical phenotype to our Tdna mutant, which is good news. So I'd like to acknowledge the people who did the work. I do the work myself in collaboration with Hui Ping. I hired him several years ago. He did the P expansion transformation and the Tdna mutant construction. He's an agrobacterium transformation expert. He's now at UC Davis. Vernetta Gaskins is my support scientist. The Cooper lab, Brett Cooper is a mass spectrometry proteomics wizard, along with Dr. Gary Bouchon and Joe Mowry that did the SEM, TEM work. From Nancy Keller's lab, Denarius Luciano Rosario, who helped generate the blistering 1 deletion mutant, who's a student, PhD student in Nancy's lab. And then the funding, which is almost entirely from base funds provided from USDA ARS National Program 303 Plant Disease. But I'm also funded by USDA, NIFA, Crop Protection Pest Management, and I have competitive funds from the Washington Tree Fruit Research Commission. And with that, I'd like to end my presentation and take any or all questions you may have at this time. And again, I really appreciate the opportunity to present this and talk about my science with you guys. Thank you.
Thank you, sir. Thank you for wonderful lecture. I hope audience must be having so many questions in their mind. I request the audience to interact with Dr. Vaine. Please.
So, it's a very elaborate presentations, and we are really thankful. But I have a couple of queries with reference to your when you just create mutant using Tdna mutagenesis, and you compare with your wild type. It was not because the mutant was still virulent. So it seems that there may be certain other also associated genes that play a role in the governing virulence. So that may be one of the, you can say, areas which you can, how we are looking at that angles, particularly that aspect. So this is first question. Second one is that when you your forward gene approach, genetics approach, that clearly indicates that there may be the modulations in the germ tube, or you can say germinations behavior of the your conidia. So do you think that in the when you reverse genetics approach, when you did first one, there you could not find difference in the germinations of the conidia? So as you explained, so what is the difference and why there is a changes in the germinations pattern in mutant versus wild in forward genetics, while in mutant versus wild in reverse genetics? So and other is that PAT-E, role of PAT-E, maybe confusing 2-3 questions. Role of PAT-E is well explained, and we know that patulin is very toxic, and while escadiol is non-toxic. So can we, means you can say, target particularly that detoxifying pathways for the development of resistance in apple? Yes. Yes, why
yeah. So you want, so let me answer your second question first. So that's a good question. So if I understand correctly, the idea would be to target PAT-E so that the fungus maybe could not. So my idea is to target PAT-E. In other words, what if I was thinking about generating an RNA interference construct to be able to silence production of PAT-E so that you could not produce patulin anymore. So if you can knock out patulin production, you can basically make a weapon to shut that toxin production down in the fungus. So that's one approach that you could use. Regarding the detoxification of the compound, we don't know what's happening how the fungus deals with its own toxin, except I can tell you that efflux is important. So if any of the toxin happens to get back in or is accidentally made inside for some reason, efflux is important. So if you block efflux, you can be toxic to your own pathogen or own toxin. So that's another strategy. If you can shut efflux down, you might be able to get the fungus to poison itself, so to speak. So we have data that's just got accepted to phytopathology on this, where we show that that some of the mechanism for toxin tolerance in penicillium. So I think, did that answer your second question?
Yes, second question was that germinations changes in wild and pure mutant.
Yeah. Well, first of all, so the blistering 1 mutant's a Tdna mutant, and so whereas the knockout mutants, the gene is completely gone. So they're not the same type of mutation. So you would hope that they would be very similar, and they are, but I don't know the biological reason for the morphological difference. The germination rates are almost the same, and I have pictures. So one other thing I forgot to mention was right when I finished all this stuff, the pandemic hit. And so I was trying to get good pictures of the conidial defect and move things forward, but I haven't been back to the lab for quite a while because we're still dealing with it. But long story short, that's something that we want to investigate, is understanding what is that defect. And I think it has something to do with blistering 1's not present in that, is completely absent in that mutant. And so I think they're having trouble. I think those vesicles are having trouble getting oriented properly, and the proteins aren't being secreted. So they kind of pool, and then they find their way again, and then they start to orient straight. I think it's got something to do with that. This is a very metabolically active situation, germination. So that's the best I can answer, but I don't know is my short answer.
Yeah, thank you.
You're welcome. Thank you.
So there is one question in the chat box. As the alteration of pH is one of the feature of pathogen, is there any possible involvement of tissue alkalinization through RALF peptides? Okay.
I think that's a great question. I have to admit my ignorance here. I'm not necessarily. I don't know what are RALF peptides. And let's see, alkalization. I can tell you that in general, the fungus will acidify. So it dumps acids into the tissue and acidifies. And this particular fungus doesn't like alkaline pH so much. So that's why the mutant also doesn't create as much decay because of that. But so you're saying maybe there's more accumulation of these RALF peptides in the mutant, perhaps? Is that what you mean? And what are RALF peptides? Can you tell me? I don't know. Dr. Sridhar. Yes.
So there's one more question. RALF peptides are rapid?
Yeah, RALF peptides are rapid alkalization factors of the host. If activation or silencing of this process is influenced by the pathogen, that's what my interest.
Interesting. Very cool. I don't know. Great question. I don't know. That could definitely be something I could look at. If they're there, if we've detected them, they should be in my proteomics data set. I need to look into
These are very small peptides of 18 amino acids or something like that, which has been implicated in the pathogenesis of quite a few syndromes. Probably, I think, which of information, if it comes, it would be interesting to know about them. Thank you.
Yeah, that's very cool. Thank you for that.
Another question in the chat box. What type of enzyme is produced by penicillium expansion to cause rotting in there?
It's a good question. So I can tell you, well, based on our proteomics data, there's lots of enzymes produced. So polygalacturonase, pectinmethylesterase, proteases. If you go back to when I started in the lab years ago, doing column chromatography and old-fashioned enzymology, the main enzyme that I was able to isolate from rotted apple tissue is polygalacturonase, and that's the only enzyme that you can take, purify, and add to apple tissue and watch it degrade the tissue. Okay, so I'd say that one of the main factors for destroying those cells is polygalacturonase, and of course it's a concerted reaction. There's a lot of other things going on, but from a proteomic standpoint, it's very complicated. There's lots of cell wall degrading enzymes, but from a very simplistic enzymological standpoint, when we were looking at purifying proteins, we were getting mostly PG, some PMEs, but mostly PG.
I have some questions. First is, you told that blistering one mutant locus is required for the secretion of cell wall degrading enzymes, and which produce patulin, which is needed for apple infection. And in one cycle, you told that from that patulin formation occurs outside the cell of the penicillium expansion. So is it by default, or is it true for all the kind of the toxins? They initiate their formation inside the cell and they get toxic once they come outside of the cell?
It's a good question. So I think I just want to clarify. So the blistering one mutant still secretes enzymes, but it secretes them improperly. So you get some, but it doesn't secrete them properly, and then it doesn't acidify so that they work properly. You still get some enzymes being launched, but it's not necessarily the correct ones or ones like wild type. So it's gray. It's fuzzy. But you're right on the money with the patulin. So in general, this has been an emerging trend in fungi. So for example, other systems, fusarium with deoxynivalenol, DON. I don't know if you're familiar with DON toxin, but it's basically the fungus uses endosomes, and it puts the precursors and the enzymes in endosomes and then moves them outside the cell, and then the enzyme produces the toxin outside the fungus. So it's a way to it's sort of an auto resistance mechanism. So this has become sort of a theme in plant pathology, toxicology, that the fungus uses these more. This is a little bit more simplistic, but this is a theme that these enzymes, now these toxins are secreted. Now what happens to, what I didn't tell you is that penicillium has the capacity to produce like 60 or 70 different small molecules. There are other toxins, right? There's lots of other toxins, but patulin's the only one that's regulated. And I don't know what's going on with citrinin, penicillic acid, clavulin. I don't know what's going on with those other ones. So it could be similar case, but I'm not sure.
Okay. Sir, one more thing.
Yeah.
Host pathogen microbes interaction involves many secondary metabolites, right? So how these secondary metabolites fluctuate this biological interaction, means occurrence of a disease, severity of a disease.
It's a good question. So you mean like what is the impact of, let's say, patulin on other pathogens?
Yeah, that is what means you mentioned. Means like penicillium expansion, we are mutating it, we are interacting it with the host, and there will be some disease severity reduction or expansion in the pathogen. So there must be involvement of some secondary metabolites also.
Yeah, so patulin itself causes, symptoms on the fruit, so it causes cell death. You can still have you can still have infection if you have a completely pat minus mutant. People have done this. The role of other secondary metabolites in infection, I'm not sure of in my system. I'm trying to think. Oh, gluconic acid is one that's important, I know, in penicillium. I'm trying to think of some others. Off the top of my head, I don't know.
No issue, sir. And one more interesting query came to my mind after listening.
Sure.
As we know that penicillin, it comes from Penicillium species, which is a first antibiotic. In the same way, is there any mycotoxin you may have come across as you are doing a lot of work on it, which can be used as an antibacterial agent?
Yeah, so that's another good question. So patulin was initially discovered. So patulin was initially discovered as an antibacterial. Okay. Recent work that we literally just had it conditionally accepted. We showed that patulin affects other fungi. So typically, when you find penicillium infection in an apple, it's not mixed with other things. So we tested Alternaria, Botrytis, Calototricum, and one more, I think Fusarium. And we were able to inhibit the germination and growth completely of those 4 pathogens with patulin. So patulin is impacting other fungi in the infection court.
Any bacterial pathogen?
Bacteria, it impacts E. coli, I know. I haven't looked at other ones, but that's a good question. I'm not sure.
Okay, thank you, sir.
Sure.
In audience, we have Dr. Anupam Verma, sir, is there. So please, I request you to interact or say a few words.
I'm very much here.
Yeah, yes, sir.
It has been a very nice lecture and very important.
Thank you.
In fact, there are lots of problems. I can only say that I started working on this problem about 60 years ago. So yeah, it is of my interest, but I'm very happy to see the development which have taken place.
Thank you.
Only question I want to ask you, what is the practical solution to avoid such contamination or infections?
Yeah, that's a good question.
You are saying every question a good question. It's not a good question.
Yeah, no, it is. It is because I didn't really touch on that much. So the practical solution to this would be, I think you need to come up with a rot-resistant cultivar that you can integrate into your pest management strategy. One of the things we're working on is trying to understand where the inoculum comes from in the packing house and how to combat that. So the practical solutions, I think, short term would be identifying where the inoculum is and combating that. And then I also think developing the rot resistant cultivar that you can basically use to help you fight this thing. Of course, you could go the chemical route and maybe some natural products or biocontrol products. You don't have a lot of tools, but from my standpoint, I think those would be the things that could be done to practically. What's your opinion on what was your goal?
Major difficulty when is that to develop a cultivar of a tree species takes a long time.
Yeah.
Even if we have found sources of resistance or resistance in a particular selection, a selection one you can go ahead and do mass multiplication and go ahead. But developing a cultivar takes time. And this is actually a market-related problem. The only thing is that we do not yet know completely how much it is affecting the health of people, number one, working in those storehouses where the infection is common, just as the lung disease, in the silos, and how much this infection is causing. And number two, what happens when people inadvertently take, you said cider, it could be used in cider and goes on. But is there any study to say that such a cider is really toxic?
I don't know the cider literature.
I tell you what, I will send you my email. Because I don't want to take away discussion time from others.
No. Well, just let me answer quickly.
Yeah.
I don't know the cider literature as well, but I can tell you that fermentation does impact patulin levels better than anything else, but it doesn't get rid of. I think the best I ever remember seeing was like 95% reduction.
I mean, how much of the toxin goes away?
About 95%, but still there.
Yeah.
I really appreciate your questions and insights. I'd like to talk to you more.
It will be a pleasure. It will be a pleasure talking to you. As I said, it's my love. I started my research with this problem.
Yeah, it's great.
Problems. Okay.
Let's see. Dr. Sridhar says, as the resistance source is scarce or not available, probably if more knowledge is gained about the process of pathogenesis at genome level, probably manipulating resistance through gene editing would be interesting. That's another good point.
That will take time.
Time.
And by the time you do it, the things have moved further. It should be done. I don't say it should not be done.
Yeah.
In fact, I worked on tree disease as well. And somebody suggested many years ago, have a resistant cultivar. You see, the cultivars in the case of fruit trees, I'm just making my point and others can counter. I'll be happy if somebody counters it. You see, for example, apple. Apple, you like particular variety much more than the other. The Chinese apples have come up with some varieties which are liked by some people. But still this green nice apple which you have, which is actually being used more as a pollinator, is tasty, isn't it?
Yes.
And it's a Washington apple, very crisp and tasty.
Yeah.
Now, since you have been working on apple, I want to ask you a question and tell me if you can help in this. You see, the storage of apples in transit is very important. So, are these being stored in nitrogen environment or some other mechanism?
Well, I think you asked two questions in one, because you said transit. So first of all, cold chain management is important when they are being moved long distance. However, the majority of the large scale packers are going to or already have what's called control atmosphere storage, which is cold down to 1 degree Celsius, from 1 to 4 degrees Celsius, depending on the variety, and 1% CO2, 1.5% O2. So low oxygen, low CO2 concentrations, and adding 1-methylcyclopropene to inhibit apple fruit respiration. So there's very sophisticated storage available. So that being said, and you even under those conditions, you're still having decay. And what we're finding at the preliminary level is that by looking at packing houses over time, a lot of the spores are floating around in the air and they're on the bins. And so I'm trying to get the producers to sanitize their bins, and they're telling me they don't want to spend the money doing it because they don't think that that's going to really help them too much. And I've got to do some research. I've got a grant to try to show if sanitizing the bins helps. And that's one of the things we're working on. But again, that's something else I haven't really made much movement on.
Thank you very much. It was wonderful interacting with you. And
Good,
wish you all
good
the
talking.
best. And we'll
Thank
be
you very
in touch.
much.
We'll be in touch.
Thank you. Yes, sir.
Thank you, Dr. Vaine.
Thank you, sir. It was a very good session with lot of interaction. And now I request Dr. Kalyan Mondal sir to thank the speaker and all the other participants. Dr. Mondal sir, Kalyan Mondal sir.
Please unmute, sir.
Yes, thank you, Sangeeta. And we really had a very, very elaborative, interesting talk. And Professor Vaine has made it very interesting with the interaction session also, because a lot of questions we had in our mind, and he categorically answered all these queries. And we are really thankful, Dr. Vaine, because this is the early morning there, and I think it's really we had got enough time from your side, and we had got very good insight into this blue mold biology. Particularly, it is a very interesting that you have intersect all the aspects of how a mutant behave compared to wild, and it's gave us a lot of insight into the recent tools, how morphologically as well as molecular level as well as proteomics level, what are the changes happen in wild type and mutant that you categorically explain. And it is a nice to have you with this IPS talk series, and we'll be looking forward to interact with you in future IPS series if you have. And with this, I'm also thankful to Professor Verma for your interactions and for your presence here in this talk. Also, Professor Sridhar sir, and our President Madam Pratibha, and all other dignitaries, including Secretary Dr. Robin Gogoi, and all other IPS associated dignitaries. And I think also Dr. Dilip Lakshman. So I think it is a very good that we are discussing on this important talk series through IPS webinar series, and a lot of informations we are gathering from our students as well as our other interesting colleagues, faculty are also getting enriched through this talk. And with this, I once again I extend thank you from Indian Pathology Society to Professor Vaine for his interesting and elaborative talk, and to all the participants for making this talk series a grand success. Thank you very much. Good evening. And
thank you. Thank you so much. I appreciate it. Thank you for the opportunity.
We extend thanks to Dr. Dilip Lakshman sir also. He is a bridge between Dr. Vaine and IPS. He brought him here. So thank you, Dr. Dilip Lakshman sir.
Yes.
Yeah. Thank you. Thank you.
Let me also say that Dr. Bharma is my teacher. He is the virology professor. And I'm very happy that Dr. Bharma is very active till now. So once again, my obeisance to Dr. Bharma.
Nice seeing you, Dilip.
And thank you very much for, just one request you agreed. And I know that you have been having a morning walk now, and your other activities you set aside. So I'm very thankful. And I'm also for the Platinum Jubilee. And, all the dignitaries, my obeisance.
Thank you so much. I appreciate the opportunity. It was a pleasure.
Thank you, sir.
Yeah. Thank you, Dr. Vaine. Yeah.
Yes, sir.
Thank you, sir.
Yes.
Yeah, Dr. Vaine. So I congratulate you for delivering a nice talk. So myself, Dr. Robin Muber, Secretary of Indian Phytopathological Society. So today, right from early morning, I think you have you had a good start for the day activity today.
That's right. I
Yeah.
did.
Okay. So definitely we'll be keeping touch with you for
Yes.
our future activities.
That'd be great. I appreciate it.
Okay. Thank you. Thank you.
All right, guys.
Thank you.
So,
Okay.
is there any way that I could get maybe a link to any future talks to listen to the other talks on the IPS circuit?
Yeah, we will send you the link. There will be this weekly. We will be having at least 2 talks. So
Okay.
we'll make sure that the links will go to you.
I appreciate it. Yeah, thank you.
Only thing that the timing will be different. It may come
okay.
to late night for you, but
That's
So
okay.
let me tell you that Amy Rossman, she will give a talk, and Kathy Aimi. Kathy Aimi probably you know also, right?
Yes, sir.
She was in our institute, but then she left. And also someone in nanotechnology from Connecticut, he will be also presenting.
Nice.
So they will let
Dr.
me
Wade
know.
Elmer. Dr. Wade Elmer.
Oh yeah, I know Wade. Yeah.
He will be presenting on nanotechnology.
Nice. Yeah, that would be great. I'd like to hear I'd like to hear
And
a variety of the talks.
Most welcome.
And so next time, when all this COVID thing will go down, maybe when should visit some of the institutes in India and interact and collaborate with them with your expertise and their practical suggestions. Maybe it will be very easier. So
That'd be great.
this COVID situation, it brought us together digitally. That's something interesting.
Yes.
But you should visit India sometimes.
I would like that. Thank you very much.
Most
Thank you.
welcome,
Thank
sir.
you.